Okay, everybody, this is going to be our presentation for fermentation and cell respiration. The first thing I'd like to do is I'd like to point out a few things. These are the notes that I used in class, and the thing I wanted to do is start previewing the information that we're going to find inside of our slideshows a little bit more easily. This helps you organize your thoughts, and it helps it a little bit easier to navigate when you're trying to learn new ideas. The topics that we're going to be covering today are going to be aerobic and anaerobic, what those terms actually mean, how they're different from each other. This will actually play a big role later on. We're also going to compare photosynthesis and cell respiration. Now, this should be really important because we already learned a good deal about photosynthesis early on. If you don't remember anything, you can go back to the previous presentation that we went through. Aerobic respiration process, we're going to learn about that. We're also going to learn about the two processes that are really going on, or the two systems that are taking place in there, called the Krebs Cycle and the Electron Transport Chain. And then we're also going to talk about the anaerobic respiration processes known as fermentation. There's two of them that we're going to focus on. Those two are going to be lactic acid, and the other one is going to be what we call alcohol fermentation. So we're going to really focus on those two versions, even though there's more than that. So at the end, you'll also find a summary, and we'll try to make things work from there. All right. So like I was saying, cell respiration, it's going to be the chapter that we've been working on for photosynthesis. That's chapter 4, but we're going to pick up now on section 4 and section 5, and then we'll divide that into another portion, which will be section 6. So, cell respiration. This is going to be an extremely important process, not just to animals, which have only mitochondria, but it's also going to be an important process for plants as well, because they contain mitochondria as well. If you remember correctly, we said plants contain a special kind of organelle called a chloroplast, and the chloroplast is responsible for making sugar. Well, that sugar eventually needs to get broken down, and we need to make some energy from it in the form of ATP. In order for that to happen, oxygen needs to be present, and this process is called aerobic cell respiration. There is a process that doesn't require oxygen. There's a few processes, and that's what we call fermentation, also known as anaerobic respiration. Now, this process is almost a mirror image of photosynthesis, and we'll talk about some of those similarities here in a little bit, but I just want you to recognize the beginning of one is the end of the other process. So the beginning of photosynthesis is the end of cell respiration, and the beginning of cell respiration is the end of photosynthesis. I'll show you an image on the next slide that makes a lot more sense from that. This process also, it has two stages, just like what we see in photosynthesis. They're different from it, but they have some similarities, and we'll talk about those here in a little bit. And it also takes place in the organelle called the mitochondria, which once again is kind of like the alternate brother of what we call a chloroplast. So let's check it out. All right, so the processes that we see here, they're going to be cell respiration and photosynthesis. Photosynthesis should look very familiar to you because we spent a good deal of time already talking about it. You should already have this formula committed to memory, but just in case you don't, it's carbon dioxide and water, those are and light actually. Those are the processes that we consider the reactants of photosynthesis. They are used to help make sugar and oxygen. If you know that formula, you're sitting pretty nicely, because basically sugar and oxygen are going to become the products of photosynthesis, but also they'll become the reactants of cell respiration. So basically, organisms that use cell respiration take sugar and take in oxygen. They break that stuff down and move it all up, or reshape it and all that. And they basically create energy in the form of what we call ATP. And sometimes heat is released as a process of that. And we also make some carbon dioxide and some water. Sorry I couldn't squeeze water on here, my bad. But anyway, cell respiration is pretty darn important because the byproducts, the end products of cell respiration, actually become the reactants that we find in photosynthesis. So go ahead, guys, remember, we have the reactants and we have the products. The products of one become the reactants of the other. And that's how we have the relationship between cell respiration and photosynthesis. So without plants, we probably would not have a very effective animalian system where we rely on cell respiration. And without animals, really, photosynthesis wouldn't have the things it really requires too easily. So it's kind of nice. Thank you, trees. I'll help you, you help me. So there's some major differences that we want to be able to recognize between cell respiration and photosynthesis. I tried to do you a favor and leave some check boxes here so you can kind of check off to make sure you understand things. If I look on the left side, cell respiration, it takes place in the mitochondria, and it makes a lot of ATP. If I remember correctly, I believe it's 36 ATP. It might be 34 to 36. It might even be up to 38. I'm sorry, I can't remember off the top of my head. I should have done my homework before I started talking. It's also used to make carbon dioxide and water and energy, which we saw in the last slide. And it's made up of two stages, what we call the Krebs Cycle, also known as the Citric Acid Cycle, and the Electron Transport Chain, which is related to the process of oxidative phosphorylation. You're not going to have to memorize that unless, of course, you're in a AP biology class. Now, in photosynthesis, the process takes place in something called a chloroplast. Chloroplasts are these organelles called plastids, and they kind of act like a mitochondria. They're pretty unique in the sense that they absorb sunlight, but they do kind of act like mitochondria in the sense that they do generate energy inside of cells. They make a little bit of ATP. If I remember correctly, it's something around 6 or 7 ATP. It's not a lot, not nearly as much as cell respiration. It's still very important, but it's not nearly as much. It's used to make sugars and oxygen from energy. So we're actually using energy from the sun to actually convert that into sugars and oxygen. And it has two stages. You'll remember light dependent and light independent, which we call the Calvin cycle. Just if you want to review that stuff, go ahead and go back to that last presentation that we talked about photosynthesis on. So what we're going to see here is a nice summary. It's a good image that really simplifies the processes that occur during aerobic respiration. If I look on the left side here, I see this green bluish looking box. I keep saying blocks. Box that says glycolysis. Glycolysis is the process where you make glucose, a 6-carbon molecule, and you convert that into 2 3-carbon molecules. And then we can separate those pyruvates from each other, which, by the way, they're called pyruvates, also known as pyruvic acid. These are going to be broken down in the Krebs cycle and reused, and we're going to actually make some energy there. Glycolysis, you'll notice out here, actually takes place in the cytosol, which is the cytoplasm. So it happens outside the mitochondria. It also has the added benefit of carrying some electrons through NADPH all the way over to the Electron Transport Chain, which is at the end of this process. Down here at the bottom, you'll be able to see it does make some ATP. As a matter of fact, it makes exactly 2 ATP every time it works, which is a good thing because it allows us to have energy and to have the molecules that we want for this process to occur. So without this process, nothing else would happen. That means glycolysis must take place at the beginning of all forms of respiration. It even becomes pivotal in fermentation, which is anaerobic respiration. Next up, once we have those pyruvates made, they start to move through that membrane of the mitochondria. Once it's moving through that membrane, we go through a process called the Krebs cycle. And basically, that's when we're converting our pyruvates into something called acetyl coenzyme A. That gets cycled around, and we reshape it into a few different shapes. And basically, we're making a little bit of ATP. Once again, not very much, but we are making about 2 ATP in that process. We also have the added benefit of charging up some electrons thanks to NADH and FADH, and they're going to pass on into the electron transport chain and oxidative phosphorylation. I should point out to you that the electron transport chain happens in the mitochondrial membranes here, and it's a lot like that process that we learned for the photosystem 2 and photosystem 1, the light-dependent reactions that we see in chloroplasts. But the added benefit of this version is that it is able to make a lot of ATP. I believe our number here is going to be 30 2 ATP. That is a ton of ATP, and your body loves it. And remember, this process is happening kind of all the time, every second, in all of your cells that have mitochondria, which is all of them. So you're creating tons and tons of ATP all the time, even though if you're just sitting there watching a PowerPoint, listening to Mr. Olson drone on, you could say you're doing nothing at all, or you could say your body's making a ton of energy thanks to oxidative phosphorylation, which just makes you sound super smart. So here's something to remember. Let's go ahead and define what aerobic and anaerobic respirations mean. Aerobic respiration is the process that requires oxygen in order to take place. If you've ever heard of somebody doing an aerobic exercise, they were probably breathing pretty hard because their body required more oxygen. We use that phrase to just determine things that really need oxygen to take place. Anaerobic means not aerobic, therefore it means you do not require oxygen, so no oxygen takes place. And the cool thing is we have a lot of organisms that have adapted to survive in anaerobic environments, and we kind of utilize those in different ways. Glycolysis, this is a really important term. We just kind of talked about it on that last slide. And it's the process that splits glucose, a 6-carbon molecule, into 2 3-carbon molecules, and it also makes 2 ATP. So this is the process that must take place before cell respiration can actually work or do anything. And this actually works for both anaerobic and aerobic cell respirations. Okay. So let's go ahead and break down the process of aerobic respiration into 2 parts, which we call stage 1 and stage 2. Stage 1 is known as the Krebs Cycle. I'm pretty sure that should actually have a little apostrophe above the S. And it's also called the Citric Acid Cycle. We'll talk about why it got its name here in a second, but I've done my best to keep this as absolutely simple as possible, and if you actually study these processes in depth, you'll realize I'm just grazing the surface. I'm hardly talking about them really at all. Anyway, so this stage, its job is that it's going to produce energy molecules that are later going to be used in the electron transport chain. If you go back to that image that I showed you earlier, just 2 slides ago, you'll be able to see the energy from the Krebs cycle does get used. It helps transport electrons to the electron transport chain in order to make a ton of ATP. So step 1 here, you got to recognize that these 3-carbon molecules from glycolysis are going to get broken down. There's a special molecule that's going to be involved, and it's called acetyl coenzyme A. That's a co with a capital A at the end. You'll see that picture coming up here in a second. This is going to be a really important molecule. By the way, it's broken apart using enzymes. This molecule will eventually get added to another molecule that's a 4-carbon molecule, and then we'll start adding those together and start tearing it apart. And every time we tear it apart, we'll start to get a little bit of energy that we can save it for later. A small amount of ATP is going to be made here. This ATP will be used for the second stage. That's the electron transport chain. And carbon dioxide is going to be given off as a byproduct here. So in the next image, I want you to take an eye and keep it on the process of what's happening to the carbons, these 3-carbon molecules, and what's happening to the carbon dioxide in this diagram. The first image I'm going to show you is a very simple one, and the second one is a bit more detailed, but we can still follow along with it. So this is the simple version. I like this version because it actually shows you just how many carbons are located inside of there next to its name. We see here pyruvic acid. At the top of the image here, pyruvic acid is the same thing as pyruvate, generally speaking, and this isn't always true, I don't think, but if it ends with ATE, the letters ATE, pyruvate, it's probably also known as an acid somewhere, so this is called pyruvic acid or pyruvates. Anyway, we can see we move 2 hydrogens off of here, and we break off a carbon dioxide little combination here, and since you have 3 carbon and you remove 1 from the CO2, you end up with 2 carbons left behind, which we call acetyl coenzyme A, that molecule I was talking about on the last slide. This 2-carbon molecule is going to be added to a different 4-carbon molecule, and we're going to form a new 6-carbon molecule, because 2 plus 4 is 6, called citric acid, which we also know as citrate. This is where the actual Krebs cycle gets or receives its title its second title, called the Citric Acid Cycle. If we have 6 carbons here and we go ahead and we eliminate a carbon dioxide molecule and 2 more hydrogens, we end up with a 5-carbon compound. We once again remove more CO2. We remove some hydrogens. We end up with a 4-carbon compound. We can change the structure some more, keep it a 4-carbon compound, lose some hydrogens, and we continue this process. And every time we're pretty much popping off some of these molecules, we're able to convert NAD into NADH, which can later be used in our process for the electron transport chain. So we're also going to be making a few ATP molecules in this process, which you'll be able to see in a lot better detail here in a second. But anyway, these 4-carbon compounds eventually get recycled, so we can come across a new acetyl coenzyme A that gets added to the 4-carbon compound, and we recreate citric acid, and we recreate that entire cycle once again. Now this only follows one of the pyruvic acids, one of the pyruvates from glycolysis. This really happens twice. Overall, this happens 2 times when it comes to how those pyruvates are actually handled. So yeah, this one is a bit more complicated, but I still like it because it actually does a really good job on showing you exactly the shapes of those molecules and how they transform throughout this process. So if we look at the very top of the process, from glycolysis, we end up with the pyruvate, which is pyruvic acid, and you can see it's got 1, 2, 3 carbon molecules attached to it, and then that eventually will be converted into acetyl coenzyme A, which, if you look off to the left right here on the top left corner, it's got 2 carbon molecules. 2 atoms. This will eventually be added to another molecule, which is originally a 4-carbon molecule. Hey, look, it's like the same thing we already talked about. Anyway, they'll be added together. We make our citrate, which you can see over here is a gigantic 6-carbon molecule, and then we start to play around with the structures a little bit. Basically, we just kind of shuffle things around, and when we do that, we start converting NAD to NADH. We pop off some CO2s. We're able to make once again NAD to NADH. We're also able to make a little GTP. You're not going to have to remember exactly what that is, but I just want you to know it might be a thing. And anyway, we're just going to kind of go through that process where we modify these molecules and we kind of pop off CO2. And this is where the CO2 in your exhaling breaths come from. This is the CO2 that you release as you breathe out. The second stage of cell respiration is called, or referenced as, the Electron Transport Chain, or oxidative phosphorylation. I once again am oversimplifying this process. It's a crime what I'm doing here. But stage 1, what happens is energy is being transferred to here from the Krebs cycle. So that process we just looked at is actually sending over some energy. That energy is really going to be utilized to help us absorb some oxygen. So now whenever you inhale in your breath, the oxygen from that process is now being used. We're going to make lots of ATP. Once again, earlier I think we said we make about 30 ATP. And water and heat are going to be given off as byproducts here. You can actually kind of test this idea. If you put your hand to your mouth and breathe out, you can feel you are giving off heat. If you are giving off heat off your body, we can tell that something's going on inside of us in terms of energy. And we're also giving off water. We sweat out water, we urinate water, but we also breathe out water. Yeah, even though we require it, we do still give it off as a byproduct sometimes. This image should look vaguely familiar to you. When we were talking about photosynthesis, I actually referenced the electron transport chain during that. I believe during the photosystems, we said photosystem 2, photosystem 1, which was part of the light-dependent reactions. These two processes are strikingly similar to each other. They both take place in a membrane that we find inside of the energy processing cells, or organelles inside of cells. The only difference really that we see here is Well, there's no light involved in mitochondria. They actually have those energy molecules we made previously from the citric acid cycle or the Krebs cycle. That's really going to start down here. And we're going to kind of convert some energies, use those up, and it helps us move those excited electrons along through here. And as those electrons move through there, we can see these hydrogen ions move across that membrane, and they're going to change the gradient, the concentration gradient of those hydrogen ions to one side of the membrane. Finally, when we get down to the bottom here, we can see we do make a little bit of water as a byproduct here. And once again, I told you we do have water as a byproduct. And since we see such a high concentration of hydrogen ions inside, we pass through another molecule, once again, just like in photosynthesis, called ATP synthase, which if it ends in an ASE, it means it's probably an enzyme. And this is the enzyme that helps change ADP into ATP molecules. And that happens each time one of these hydrogens move across that membrane. And that's an amazing process, which we can certainly make a ton of ATP with, and I believe we make 30. So that's just a really powerful process that really occurs there, and this is why we work as efficiently as we do, and this is why you love to have sugar all the time. So let's go ahead and talk about fermentation. This is going to be Chapter 4, Section 6. Section 6. Listen to me messing up. Anyway, let's go ahead and try this. So we already mentioned there's this process called glycolysis, and I just want you to remember this is where cells break apart 6 carbon sugar molecules into 3 carbon sugars, and it also makes 2 ATP molecules. Now this process does not require oxygen, and earlier we stated the things that happen that do not require oxygen are called anaerobic processes. They actually usually stop functioning whenever oxygen does appear. And fermentation is also really just kind of a process that makes glycolysis continue. Fermentation itself doesn't actually make energy. It kind of uses up another form of energy to help us make ATP, which can be used later in cellular processes. The example of the image that I have up here, these are actually yeast cells. These are fungi that are adapted to live in an environment with no oxygen or very little oxygen, and basically they make their energy very little bits at a time by making 2 ATP over and over and over again. And when they're doing that, all they're doing is just allowing glycolysis to continue. They do make a byproduct. We're going to talk about those 2 byproducts when we talk about the 2 different forms of respiration or fermentation that we really care about here. So normally you maintain your oxygen levels, but when you do hard work, when you actually are breathing tough and really working hard, your body needs just that little boost. It needs that little bit of extra help because you cannot possibly keep up with your oxygen levels over a period of time. If you've ever been sprinting and you noticed, you get very tired very quickly, you just can't maintain a speed, that's because your muscles are literally saying, hey, I can't do this anymore, I need to stop, or I'm not going to work properly. And basically, you tell your body to use another energy source to help give you that boost to continue to work properly. So basically, we need a different energy source so we can still function in these kind of extreme situations. So we use fermentation, or at least a version of fermentation, to continue glycolysis for energy processes. So we're going to study only 2 forms of cell respiration that are anaerobic, called lactic acid fermentation and alcohol fermentation. These are the 2 most common ones, and actually you'll be very familiar with lactic acid fermentation from just being alive. But alcohol fermentation is actually probably the most profitable biological process that's been used here on Earth. I think more money's been made using that than pretty much any other, at least by manipulating this process. I also want to point out to you that both forms of this cellular processing involve a molecule called NADPH as its energy source. It does not use ATP, it actually just produces ATP. So you use a special kind of currency to make a better kind of currency that we use all the time in our body. In class, I use the example. Inside of our body, just like in this planet, in this world, there's more than one kind of money. a different kind of currency. In the United States, we use the dollar, but in Europe, it's very common to see them use the euro. While the usefulness and value of those change over time, basically you can still do business using two different forms of currency, and you can exchange the two. Well, NADPH in this case is going to be kind of like the euro, and we're going to make the ATP kind of like the American dollar. Basically, we use a process that uses some euros to help us make more dollars, which is our really common form of money we use here in the United States. So, yeah, that's kind of the example. Hopefully that makes sense. Anyway, remember both of these processes, lactic acid and alcohol, are both going to be anaerobic processes. They do not need oxygen in order to work. So these are pretty helpful in moments where you got to make a move pretty quick, or if you're a bacteria that lives in an oxygen-deprived area, and we'll talk about a few of those. So, to continue the tradition of keeping things overly simplified, and truthfully, fermentation isn't too complex, especially compared to aerobic respiration. Here we go. We're going to talk about what's actually going on there. Number one, glycolysis makes ATP. We talked about how it actually makes 2 ATP that can be used for elsewhere in NADPH. These pyruvates from glycolysis are 3 carbon molecules. They're going to be formed, and then they're going to be basically converted from a pyruvate into what we call lactic acid, or called lactate. This is a really important byproduct that we actually see in our muscles after you work them out. If you were in class today, when I am recording this, I'm recording this on the 3rd of November. I told you one of these stupid stories that I tell you sometimes, where when I was in high school, I remember working out on a bench press for the first time in my life, and I ended up making my muscles so sore that I literally couldn't even pick up a remote control or my Star Crunch that I wanted to eat for a snack after school. Hopefully you've never been in that situation, but what was happening is I was building up so much lactic acid in my muscles, I really didn't feel like moving them or doing anything with them. This simple process is basically described here. We start off with a glucose molecule. It goes through glycolysis, which basically means we're breaking the sugar, glucose, and we are going to create something called pyruvate. As we convert this form of energy, and we're using this form of energy, what we're doing is we're turning that pyruvate into lactic acid, or lactate. Basically, it's still a 3 carbon molecule. It's still both a 3 carbon molecule, but we just kind of changed the structure. So that's all that's really happening there. If it builds up in high enough quantities, that's when we start to feel that real good pain or soreness or burn that you experience in your muscles. Sometimes it's a good thing. It gives you a good sign that you're using those muscles as you're working out. Tells you kind of if you're working out certain muscle groups that you're targeting. Or sometimes it happens at the most inconvenient moments, as let's say you're working a whisk and you're trying to make some whipped cream for your delicious pie. Maybe your arm starts to cramp up and starts to hurt because of the buildup of lactic acid from that repetitive motion. Alcohol fermentation is a lot like lactic acid fermentation, with one major difference. Basically, number one, we start off, glycolysis makes ATP and NADPH. Number two, same thing as the last process, pyruvates, these 3 carbon molecules, are going to be created, and then these pyruvates are going to be converted. But before they're totally converted, we break off a carbon dioxide molecule to make them work properly. Basically, this carbon dioxide is going to become a gas that is released. So we start to see bubbles basically form inside of our mixtures here. You see this all the time when it comes to making things like bread. Bread is pretty much utilizing yeast and basically containing their burps and their farts as they eat some sugars. And basically, you're trapping that inside of the dough, and that's what actually makes the little bubbles inside of your dough, which makes your bread so fluffy and delicious. The other thing is the part that is left behind is now a 2-carbon molecule, and that 2-carbon molecule is considered an alcohol. Most of the time it's ethyl alcohol or ethanol. This alcohol is the most common alcohol that we see all around the world. This is what we find in wines and beers and like champagne. And this is the stuff, and actually this explains exactly why champagne and beers are so foamy and bubbly. If you've ever seen somebody like after a race car competition, a race, they'll open a bottle of champagne and it goes spraying everywhere. That's because there's so much CO2 trapped inside. This simple process is really shown in this little diagram here. Once again, the top part is always the same. You have a glucose molecule. You're going to convert that via glycolysis into a pyruvate, and this is where the changes occur. The pyruvate is turned into a 2-carbon compound. It's a 2-carbon compound because pyruvate itself is a 3-carbon compound, and we just break off a CO2 molecule, and then we are left behind with a 2-carbon compound, which is basically ethanol, which is going to be those alcohols that are used in beverages as well as energy sources. So I want you guys to remember what these are actually used for. Fermentation doesn't make energy itself. It actually spins energy in a different kind of molecule, and glycolysis actually makes the energy that we care about. Fermentation just helps that process continue to occur. Lactic acid fermentation, of course, is used in your muscles, and it's utilized by some bacteria and fungi as they try to survive in low oxygen environments, like the bacteria that exist maybe inside of your gut. Alcohol fermentation, pretty common, especially by humans, what we've manipulated with it. We help make our breads, our cheeses, or our actual digestive bacteria. They can rely on this as well, which could mean that you do have a byproduct inside of you called alcohol. So we have these specialized cells inside of our livers that actually are designed to help remove that stuff. So to summarize kind of everything we've been talking about, you need to know what the beginning and end products of cell respiration are. If you remember back to that, I think that was the second slide or third slide, I showed you the two cellular or chemical formulas. You need to know the chemical formulas for both photosynthesis and cell respiration, but in this case, cell respiration, you got to know cells that have mitochondria need to take in oxygen and they need to take in sugar, and from that they make energy, they make water, and they make carbon dioxide as a byproduct. The next one here, how are anaerobic and aerobic respiration different from each other? Well, basically, aerobic and anaerobic just mean one has oxygen and one does not have oxygen. So aerobic means it uses oxygen, and anaerobic means it has no oxygen. If you know that, you'll be in pretty good shape. Do you see the differences and similarities between cell respiration and photosynthesis? Well, I hope so, because we did an entire slide where I distinguished the differences between them. Just make sure you recognize those. You should already know a good deal about photosynthesis, and now all you got to know is the small differences between them, which really, if you get down to the nitty-gritties of it, isn't too different from each other. You got to know what the two systems taking place during aerobic cell respiration are. You should remember we have that process called the Krebs Cycle. And then we also have the Electron Transport Chain, which I'm going to write here as ETC. The Krebs Cycle, I wrote Krebs. That's hilarious. Krebs Cycle, Krebs. I'm so dumb. I don't know if you're going to listen to this, but you can make fun of me and my poor spelling abilities at 11 o'clock at night. What is really happening during the Krebs cycle and the electron transport chain? Do you know what's going in, what's coming out? Do you know if ATP is being made? Is it a lot? Is it a little? Those are things that you should also be able to answer as well. What is glycolysis and how is it used in these processes? Well, glycolysis is the very beginning of all forms of respiration, and heck, even with fermentation, it's pretty much just being forced to continue, and that's why it's as effective as it is. Lactic acid and alcohol fermentation, how are they different from each other? Well, they use the same beginning process through glycolysis, and they use the same molecules that help them work, but Basically, lactic acid's byproduct, the ending product, is what we call lactic acid, which is what makes your muscles hurt. And then alcohol fermentation is, instead of making a 3-carbon lactic acid or lactate, what we end up doing is making ethanol, which is a 2-carbon molecule, and then we break off CO2. So really, you should also recognize there is an extra byproduct of alcohol fermentation, and that is carbon dioxide gas.